revision of Biology Paper 2, KCSE, that is a 2024. The first question: the following word equation represents a biological process that occurs in green plants. We have carbon dioxide plus water. We get glucose plus product E. In part A, name the cell organelle where the illustrated process takes place in green plants. And that is a chloroplast, simply because this process is a photosynthesis where the carbon dioxide combines with water in the presence of light to produce glucose and oxygen, and it occurs in a chloroplast because they have chlorophyll which are able to trap light energy, and the light energy is used to split water molecules into hydrogen ions and oxygen, and some of energy is used in the manufacturing of adenosine triphosphate, which is later used in a dark stage of photosynthesis. But B, Roman 1, give the likely identity of product E, and that is oxygen. And when writing oxygen, please avoid the chemical symbols. We give the full name, which is oxygen. Roman 2, suggest ways in which the rate of production of E can be increased. And because photosynthesis is enzyme controlled, it means the process is affected by factors which affect enzymes, for example temperature, and at optimum temperature the enzymes are most active, and it means oxygen, which is product E, will be produced more, or there will be increased rate of production. Also, we can accept increase in temperature, but the temperature should be at optimum. Beyond the optimum, the rate of production will be very low, or even no production, because enzymes are denatured by temperatures above the optimum. Another factor, optimum light intensity, also will increase the rate of production of E. Then increase in concentration of carbon dioxide, and what you should realize is that these factors we are talking about are factors which affect photosynthesis, and an increase in these factors will cause an increase in production of glucose and the product E up to a certain level where they become constant and other factors become limiting. Then another way is by increasing water availability, or the amount of water. Next question, part C, suggest one possible source of carbon dioxide if the above process was artificially conducted in a school laboratory. And artificially, we can use sodium hydrogen carbonate, which dissolves to give out carbon dioxide. Apart from that, sodium bicarbonate, or you can use symbols, but said you should use words, that is sodium hydrogen carbonate. Then part D, explain how the illustrated process is significant in nature. And number 1, photosynthesis is significant in that it leads to air purification, or in other terms, removes or reduces the concentration of carbon dioxide in the atmosphere, or regulates the amount of oxygen and carbon dioxide in the atmosphere. Apart from that, it increases the concentration of oxygen in the atmosphere, which is used for respiration or production of glucose. That is another importance of photosynthesis. Remember, the oxygen is given out as a byproduct, and it is used in respiration to yield energy for animals. Question 2. In an experiment, the number of red blood cells present in 3 samples of blood labeled F, G and H was estimated. Different concentrations of salt solution were then separately added to the samples. After an hour, the number of red blood cells present in each sample was counted and recorded as shown in the table below. The first column is the blood sample F, which was added or put in a salt solution concentration 1.0. Then the number of red blood cells remained the same. Then G, 0.6 percentage concentration of salt. Number of red blood cells, fewer cells were observed. Then H, 0.4 salt solution percentage concentration, no cells observed. Part A, name the physiological process being investigated in the experiment, that is osmosis. Account for the results made in blood sample F, and this one is F, 1% concentration, the number of red blood cells remain the same. Let's look at the account. In accounting, you state the observation, then you explain. The concentration of cell cytoplasm was equal to that of 1% of salt solution, or you can say the salt solution was isotonic to the cell cytoplasm, and therefore there was no net movement of water molecules in and out of the cell, or in other terms, it remained the same, or the cell was not hemolyzed, or the concentration of the cell cytoplasm was lower than that of the 1% salt solution, and the salt solution was hypertonic to the cell cytoplasm, therefore no net movement of water in and out of the cell, therefore the cell remained the same. Part C, account for the differences in appearance of red blood cells in F and G. This one is F, and we have G. At the end of the experiment, the red blood cells in G appeared swollen or bulge outwards. Cells became crenated since the solution was hypotonic to the cell cytoplasm. Hence, net movement of water molecules into the cell by osmosis. While in F, it was a normal shape of size since the solution was isotonic to the cell cytoplasm, hence no net movement of water in and out of the cell. Part D, with the reason, predict the possible observation that will be made if blood sample F was treated with a salt solution of 2.6%. This is F. Number of red blood cells will remain the same. The solution will be hypertonic to the cell cytoplasm. Then water molecules will be drawn from the cell, making the cells to shrink or appear crenated. Part E, say the importance of the process being investigated above during urine formation, and that is a reabsorption of water into the bloodstream from the kidney tubules, or it helps conserve water, or not much water is lost through urine. Then question 3, a genetic investigation on a certain population established that in human beings, premature baldness is controlled by a dominant gene, N, that is capital N, located on the Y chromosome. Part A, work out the phenotypic ratio of the offspring of a couple comprising of a normal mother and a father with a premature baldness. Since premature baldness is located on the Y chromosome, the cross, we cross a normal mother, and females have a XX chromosome. Then a father with premature baldness, fathers or males have XY, and this N, the gene responsible for premature baldness, will be on top of that Y. Then must have a cross. We have the phenotypes: normal mother, bald-headed father. Then the genotypes must be correct. Then the genetic the cross. Then we have gametes. The gametes should be inside this circle XX, and the circle should be complete. The same to this other side, X, Y, superscript N, the gene for premature baldness. Then this line should not pierce through the gametes. Then we have fusion. Then the F1 generation phenotype will have this one is a normal daughter, then a bald-headed son, a normal daughter, a bald-headed son. Phenotypic ratio, because we've been asked about the phenotypic ratio of the offspring, will have a normal daughter. Normal daughter will be 2. Then a bald-headed son will have 2. You simplify, it becomes ratio 1:1. That is the phenotypic ratio. So when doing the genetic cross, you should observe this cross. Then the gamete should be encircled, and the lines should not pierce through the circles. Then the correct genotypes and the phenotypes should be there. Then the fusion, and for you to get the 5 marks, the phenotypic ratio should be there. It should be indicated. On to the next question. Part B. What is the probability that this couple will have a daughter with premature baldness? It is 0, because as we've said, daughters or females, they have the XX chromosome, and premature baldness is only associated with Y chromosome, which is only found in males. Therefore, the daughters cannot have premature baldness. Part C. Name 2 sex-linked traits in human beings associated with the X chromosome. And we have hemophilia, color blindness, muscle dystrophy or Duchenne muscular dystrophy. Those are examples of sex-linked traits associated with the X chromosome. On the Y, we can have premature baldness as represented above in that question. Then apart from that, we have hairy pinna and hairy nose. Those are examples of sex-linked traits on the Y chromosome. And these ones, hemophilia, color blindness, muscle dystrophy, that is on the X chromosome. Then apart from using these lines, we can use a Punnett square. On the Punnett square, we have the genotypes, that is the female or the mother, XX. Then we have across the father, who is having premature baldness, XY, superscript N, the N which causes premature baldness. Then inside the square, we have the symbols, and this represents the male. Then the gametes, X. Then we have Y, superscript N, because it is associated with the Y. Then on the other hand, this symbol represents female, X, X. Then they fuse XX with XX, you get a daughter. Then YN with X, you get XYN, a son with having premature baldness. Then X, XX, we get a daughter. Then XYN, XYN, get a son who has a premature baldness. Then maybe a caution, the symbols should be placed correctly. For example, the male should represent X and YN, then female XX, in order for you to get the maximum max. On to our next question. During an ecological study in a lake ecosystem, a group of students made the following observations on the feeding relationship among some organisms. Let's look at it. Lobsters feed on algae. Small fish feed on lobsters, worms and caterpillars. Caterpillars feed on algae. Worms feed on caterpillar. Large fish feed on small fish. Penguins feed on small fish, lobsters and worms. From the students' observation, construct a food web for the ecosystem. And a food web simply contains a interconnection of food chains. And both the food chain and food webs, they start with the producer. And in this case, producer is the algae, because it is able to make its own food by trapping light energy and making its own food. The algae are eaten by lobsters. Remember, the arrow points to the eater. The lobsters eat the algae. Lobsters are eaten by the penguins. In this case, we told penguins feed on lobsters. Then on the other hand, caterpillars feed on algae. Caterpillars are fed on by worms and small fish. Small fish are fed on by penguin. Then worms are fed on by small fish. Then small fish fed on by large fish. Then the worms are also fed on by penguin. And when we have a crisscrossing here, we use a loop. The correct way to use it, we use a loop here in order for you to get the maximum max. The maximum max. From the food web constructed above, extract a food chain with penguin as a secondary consumer, and that is algae. Algae is a producer, lobster primary consumer, then a penguin secondary consumer. In this case, it ends as a secondary consumer. There is no other organism feeding on the penguin. Then explain how spillage of oil from water vessels is likely to affect lake ecosystem. This oil will prevent exit of carbon dioxide, causing suffocation or death of living organisms. Apart from that, it prevents diffusion of air or carbon dioxide, limiting or preventing or reducing photosynthesis, hence plant death. Then also prevents entry of oxygen, thus causing suffocation or death of organisms. It also prevents or reduces penetration of light, thus limiting photosynthesis or reducing photosynthesis. Then clogs respiratory surfaces, for example the gill filaments, thus limiting gas exchange or causes suffocation and eventually death. Before we move to the next question, maybe on this food web you can be asked which organism has the highest biomass. And in this case, biomass is the total dry weight of an organism. In this case, it is always the producers, simply because they are able to make their own food and therefore sustain the whole food web or ecosystem. That is the algae. Then the one with the lowest biomass, it is the large fish, because it occupies the highest trophic level, and by the time energy reaches to the highest trophic level, it has been lost to the environment through heat loss, which is through respiration, defecation, urination. Question 5. The diagram below represents a part of a cell structure as seen under an electron microscope. We have the pose. Part A, Roman 1, identify the structure represented, and that is a cell membrane or other term is plasma lemma or the plasma membrane. State two functions of the structure. Number one, it encloses cell contents, or you can say protects inner cell inclusions or organelles. Apart from that, it selectively allows materials in and out of the cell. We say selectively because it is semi-permeable. Roman 3 suggests two ways in which the function of the structure can be impaired. And number one, in extreme pH, and that can be caused by acids or strong bases. And apart from that, extreme temperature, that is a too low temperature or too high temperatures, simply because this structure is made up of proteins, and proteins, they are affected by extreme pH and extreme temperatures. Part B, name two structures found in plant cells but not in animal cells. We have chloroplasts, cell wall, sap vacuole, then you also have tonoplasts. Part C, what is the purpose of staining cells before observing them under a light microscope? And staining, for example, the staining we use dyes, colored dyes, and the most common example of colored dye is iodine solution used in the lab, and the reason is for clarity during observation, or making the structures more distinct or more clear or more visible. You can be able to see them more clearly, unlike when you don't stain. Next question, that is question 6. Question 6, it is about graph. It is about the graph. I think you are lacking the table, but the graph was supposed to look like this when drawn, and scale is a 2 marks. Labeling of axis. Then we have curves, identification, half, that is 1 mark. And this graph, when drawing the graph, you should observe certain rules. Number 1 rule, or the first rule, origin. You must have origin for the y-axis and the x-axis. In this case, the origins are both 0, 0. For you to get the origin correctly, you can extend these lines here, then 0 here, 0 here, or a large 0 here. Then the scale must be correct, and this scale must be workable. In this case, this scale 10 represents 10 boxes represent 10. In this case, 10, 20, 30, 40, 50, 60. Then on the other hand, 5 boxes represents 2. Then we have a difference of 2, 2, 2, 2 up to 20. Then the correct plotting must be observed, and you use freehand. Then you must label these girls and these boys, must label correctly. Let's look at part B. From the graph, determine the weight of the boys at the age of 13 years. We have on the x-axis age in years. We look at the 13th mark, and the boys, these are the boys on the 13th year. That is a 13th year, that is a 32, a plus or negative 1. Then growth rate in girls between 13th and 15th year. So the 13th and 15th year, you must look at the 13th and 15th year. 13th and 15th year for the girls. 13th and the 15th. Let's look at the 15th year. It is this one. 15th around here, that is a 41. Then 13th year is a 32. 13th year it is here 32.5. You minus, then you minus 15 minus 13, get 8.2 divided by 2. That is a 4.25 kilogram per year. Then part C, account for the trend in growth in girls between the 14th and 18th year. 14th and 18th year. As you can see, there is a faster or rapid growth, and this is a puberty or adolescence or development of secondary sexual characteristics, hence increase in growth. Then part D, apart from sex and age, say 3 other factors that affect the rate of growth in humans. We have hormones, the state of one's health, maybe the diseases, then genetic factors or heritable factors, then we have feeding habits, environmental factors, emotions, stress, basal metabolic rate. Part E, other than weight, state a parameter that can be used to establish growth in humans, and that is height. Part F, why do girls above 12 years require more iron in their diet than boys of the same age? And this is because girls menstruate, therefore they require iron to compensate or replenish for the blood lost. Question 7, explain the effects of hypothyroidism in adults. 4 marks. And the effect is that it causes myxedema or swollen thyroid glands or goiter, and this is due to the underproduction of thyroid hormone. And there is also reduced production of thyroxine hormone, which leads to overworking of the glands in human. And this is in attempt to produce more thyroxine hormone. Then there is further leads to low metabolic rate and reduced heart beat. It also reduces the breathing rates. Apart from that, causes mental and physical sluggishness, obesity, and increased body weight. Then you'll have retention of body fluids or edema, that is a swollen feet. Then part B, describe the hearing process in humans. And the hearing process, we have the pinna. The pinna is the outer ear, in short. This pinna, it is funnel shaped to collect and concentrate sound waves into the external auditory meters, or in other terms we call it canal. Then the sound waves strike to the eardrum or the tympanic membrane, and upon striking it, it causes it to vibrate and transform sound waves into vibrations, and the vibrations are transmitted to the ear ossicles, which are the malleus, incus, and steps. The ossicles then amplifies and transmits the vibrations to the oval window or the fenestra ovalis. The oval window vibrates and transmits vibrations to the fluids, that is the perilymph and the endolymph, in the inner ear or the cochlea, and the fluids conduct the vibrations. The vibrations in the cochlea will stimulate the sensory cell hairs to generate nerve impulses, which are transmitted to the brain through the auditory nerve for interpretation, and the intensity of the stimulus transmitted to the brain enables the brain to interpret the impulses as sound of specific pitches and loudness. And the vibrations in the fluid in the inner ear are displaced or dissipated or released from the round window to the middle ear. Question 8. Part A, describe how urea is formed in humans. And this urea is formed in the liver, where we have excess amino acids are deaminated, and the amino group is removed. This amino group reacts with hydrogen atom to form ammonia, and ammonia is toxic. Therefore, ammonia combines with carbon dioxide to form urea, which is less toxic. That is how urea is formed. And then part B, describe the path followed by urea from the site of formation until it is eliminated from the body. Urea formed in the liver is carried in blood through the hepatic vein into the vena cava. Then blood containing the urea enters the heart at the right auricle, then to the right ventricle, leaves the heart through the pulmonary artery into the lungs, and out through the pulmonary vein back to the heart, through the left auricle, then the left ventricle, then it leaves the heart through the aorta into the kidneys via the renal artery to the afferent arteriole, where ultrafiltration occurs, resulting to formation of the glomerular filtrate, which enters the Bowman's capsule into the proximal convoluted tubule down to the loop of Henle, that is a descending loop of Henle, then to the U-bend, then to the ascending loop of Henle, then to the distal convoluted tubule. Then this urea enters the collecting duct. Now it is called urine, then into the pelvis, then ureter, then to the urinary bladder, where there is a temporary storage, and from where it exits the body through the urethra. That is how urea, or it is the path followed by urea from site of formation, that is the liver, up to where it is eliminated through the urethra. That was a KCSE 2024. And before I exit, maybe let's talk about this paper 2. And the paper 2 has 8 questions. The first 5 questions, each 8 marks, as you've seen. Sixth question, the question on graph, drawing of the graph, and answering questions related to the graph, that is a 20 marks. Then question 7 and 8, we choose one. There is a questions, each 20 marks. And these questions, basically the geography of paper 2, we must have a question on genetics. For example, is the one on genetics, this question. Then another question will have experimental biology. For example, this one is experimental biology, talking about an experiment. Then we have a question talking about maybe an organ or the cell organelles. Then question 6 is about the graph, and for you to get the graph, you must be able to follow the rules I've said. Then essay. For essay, you must be able to have enough content in order for you to score high marks in essay. Thank you. I'd like you to subscribe to my channel for more information and for more videos. Please kindly share and tag other people. Thank you.